cause a lot of trouble. They can cause DNA damage, cause misfolded proteins, and actually cause cellular death. And so what resveratrol does is it comes in and it attenuates all that damage that may be caused by those reactive oxygen species. And so in the literature, there has been so many different studies looking at resveratrol in different contexts of disease. And you can see by these titles, it protects against metabolic disease and Alzheimer's, and improves learning and memory, health and survival. And you don't need to be an expert in any of these fields or even a scientist to read these titles and realize that resveratrol is a potent therapeutic that can be applied to many different contexts of disease. And so I did some preliminary work as a part of this project. And this is an example of one of the many images of cells of hippocampal neurons that I analyzed as part of these projects. And so what I did is I administered different doses of resveratrol in the context of oxidative stress. So I administered hydrogen peroxide, identical to the hydrogen peroxide you find in your bathroom cabinet, and I administered different doses of resveratrol. But what I found is that it was able to influence the activity of many different proteins in the brain, including one involved in insulin signaling, one involved in learning and memory and oxidative stress, and also one involved in metabolism. And you can actually keep an eye on this one up here, PA and PK. We'll actually talk a lot more about that in my future work. So after an extensive literature review and all of those preliminary studies, I came upon my hypothesis for my senior thesis project, which is that a chronic resveratrol administration will be able to ameliorate the metabolic and cognitive deficits associated with eating a high-fat diet. And so a little bit about my methodology. We had a total of 40 rats in this project, and there were 10 rats assigned to each condition. And so this was a 2x2 factorial design study, and we had a vehicle and resveratrol treatment groups, and then we had control or chow diet and high-fat diet as a diet condition. And this generated 4 different groups of animals, again, 10 in each group. And so we have the control vehicle group and the control resveratrol treated group. And then there's also their high-fat diet counterparts, so the high-fat diet vehicle group, and then finally the high-fat diet resveratrol group. And I'll be using this color scheme as well as these abbreviations throughout the rest of my talk. So we injected resveratrol at a dose of 20 mg per kg, and that was determined by doing a meta-analysis of many of the different studies that had also used resveratrol in their studies to look at different things. We also took body weights and food weights throughout the project to monitor how the rats were growing and how much they were eating. We maintained them on these diets and treatments for a total of 20 weeks. Then we trained and tested them on various different behavioral tasks. After the testing on behavioral tasks, we sacrificed our animals and we collected many different tissues, including one of which was the hippocampus, and that's this area of the rat brain right here. And that is involved in learning and memory, particularly spatial memory. We also collected blood samples and weighed their epididymal fat pads, and that was just a secondary measure of the fat accumulation that they acquired throughout the project. And then we conducted a tissue analysis through Western blotting, and Western blotting is just looking at different levels of protein that would be in the brain. It separates them out and makes a nice way for you to measure how much there is. And so here's an example of a Western blot that I conducted, one of the many that I made throughout the course of my study. And on these blots, I looked at different markers of learning and memory, of insulin signaling and metabolism, also oxidative stress, and various transporters and receptors that are normally found in the brain as well. And all of my Western blot data is shown as relative optic density, and all of the proteins are normalized to beta-actin, in case you were interested. So what do we find? Well, resveratrol had no effect on body weights. As you can see right here, the chow diet type group showed a normal average growth curve for rats of those particular ages. And the high-fat diet group showed an accelerated version of that same growth curve. And resveratrol had no effect on any of this. It was just a significant main effect of diet. And then we weighed their fat pads as a secondary measure. So right here you can see the chow diet group. They had normal levels of fat accumulation, and there's no difference between these two groups, indicating that resveratrol had no effect. And again, the high-fat diet group showed significantly higher amounts of fat accumulation than their chow counterparts. Again, cementing this fact that resveratrol had no effect on their weight gain. Also, we are a diabetes lab. We are interested in metabolism, and so we did measurements of blood glucose. We measured blood glucose after fasting animals for an hour with a simple tail prick. And what we found is nothing. There are no significant differences saw the characteristic increase in blood glucose, and then it came right back down to baseline and evened out. And again, there are no differences here as well, indicating resveratrol had no effect on blood glucose or diet had no effect on blood glucose. So now comes the behavioral aspect of my project. So the first behavioral task we conducted was an open field task. And this consists of a rat being placed in a four-walled clear plexiglass maze, and they're allowed to explore this maze freely for a total of 5 minutes. And the way that this maze works is that rats, evolutionarily, they don't like to go out into wide open spaces. You know, if a rat goes out into an open field, it's much more prone to be picked up by a hawk and eaten for lunch. So it doesn't want to do that. So it normally sticks to the edges of this maze. However, rats are also kind of inherently curious creatures. So they will kind of check out what's in the middle of the maze just to see if it's safe. So rats that show no anxiety, they're really comfortable with their surroundings, they'll go around the center, edges, but they'll also spend some time in the center. They show really no preference. Rats that have extreme anxiety, they're very, very anxious, they will stick strictly to the edges of this maze and spend very little, if at all, time in the center zone. And so time spent in the center zone the more time spent in the center zone is indicative of less anxiety, and vice versa. So what we found is that in the chow diet group, resveratrol actually significantly increased anxiety behavior in these rats, as you can see by less time spent exploring the center of the maze. Resveratrol treatment completely ameliorated that anxiety. It brought them back up to baseline and made their performance on these tasks comparable to controls. The next task that we used was the conditioned contextual fear task. And the way this one works is that on training day 1, you place the rat in a spatially unique context, and you let them habituate, get used to this area for a total of 30 seconds. And then you give them a tone, and you give them a shock. It's a 0.8 milliamp foot shock, it's very mild. And you do this a total of 3 times. And that makes them have a fear memory of this context. They're afraid, they don't like getting shocked too much. So they're afraid of this context. 24 hours later, you put them back into this context and you measure their freezing behavior. So rats, when they're scared, they show very distinct freezing behavior in this context. And so more freezing behavior is indicative of a better memory of this context. And so what we found with this task is that the high-fat diet group showed an impairment in performance in this task. They did not freeze as much, indicating they did not remember that this was a fearful context. And this is not surprising. We've seen this result in our lab before and in other lab studies as well. But resveratrol was able to completely ameliorate that deficit caused by the high-fat diet. And they performed, again, comparably to controls. So I know I told you I did a lot of different molecular analyses as part of this study. And for time's sake, I'm just going to present 2 of those to you today. So the first one is this protein called AKT. And it is a central signaling molecule in cells to promote cellular growth and cellular survival, but it's also a very important molecule in insulin signaling. And so what you can see right here is that both the high-fat diet groups showed significant decreases in total expression of this protein, indicating that despite the fact that their blood glucose might be not different because of resveratrol or because of the diet, this might indicate that they are on the road to metabolic dysfunction. And then the control groups are both the same and both at normal levels. Finally, here's that PA and PK again. What I found was actually very interesting. So AMPK, or AMP kinase, is a molecule in the cell that responds to different changes in energy. So when the cell is in a very low fuel state, this becomes activated by adding a phospho group onto the protein. And that's exactly what I measured. I measured the activation of this protein, which usually occurs in a low fuel state. So what I saw was that in the chow diet group, resveratrol increased the activation of this protein. This is not surprising. We know that resveratrol is an AMPK activator, and so this all makes sense. Which does not make as much sense is that in the high-fat diet group, it also showed increased PAMPK levels. And in other studies using diabetic rats, they found that AMPK levels were significantly lower in high-fat diet rats. So something that we're currently thinking about in the lab, that this might actually be a protective measure, since my rats were not diabetic. They were just being fed a high-fat diet. So this might be a protective measure before the onset of any sort of diabetic pathology. And then finally, in the high-fat diet resveratrol group, the PAMPK levels were much lower, and again, comparable to controls. And this way I like